COVID-19 pandemic has made a significant impact on the health of the world's human population since its discovery in December 2019. Genome sequencing of SARS-CoV-2 or Severe Acute Respiratory Syndrome Coronavirus 2 has recently become one of the most important components of pandemic response. Knowledge about the viral genome has provided us clues to understand the mechanisms of viral infection, and it has paved the way for the development of vaccines and diagnostics. It has also allowed us to understand the source of the virus and its transmission dynamics within communities and around the world. The emergence and persistence of multiple SARS-CoV-2 variants of concern across the globe gave rise to additional challenges in existing public health interventions aiming to control the pandemic. While considered to be a normal phenomenon, the continuous virus mutation supported by sustained virus transmission in many countries can further increase the variants of concern. This potentially affects the clinical severity of cases, virus transmissibility, diagnostics, treatment, and vaccine development. These scenarios further highlighted the need for SARS-CoV-2 genomic sequencing strategies in line with the efforts to strengthen other existing public health control measures. RITM has embarked on 2 research projects to sequence the genome of SARS-CoV-2. Firstly, genetic characterization and metagenomic analysis of respiratory samples from COVID-19 cases in the Philippines. And secondly, implementing genomic surveillance to support SARS-CoV-2 control and mitigation strategies in the Philippines. Both studies will use next-generation or third-generation sequencing technologies to sequence the whole genome of SARS-CoV-2. Both projects also aim to capacitate RITM, DOH subnational laboratories, and UPNIH with the necessary equipment and skills for sequencing SARS-CoV-2, enabling rapid and efficient support to local and regional pandemic response. We will be employing 2 sequencing platforms, Illumina and Oxford Nanopore sequencing technologies. The Illumina sequencing platform uses a sequencing by synthesis method to spell out or read the genome sequences and is built for bulk sequencing. The Oxford Nanopore technology uses a pore to read the genome sequences and is designed for portable and rapid, low-throughput sequencing suitable for quick and rapid results. The results from the sequencing runs will be analyzed using a bioinformatics software. Data and analysis will be laymanized into reports that will be submitted to local, regional, and national health authorities. Here we see a timeline of the sequences from RITM samples along with sequences generated from other sequencing labs nationwide. The few lineages from the first half of 2020 were replaced by several lineages. This implies that there were multiple introductions of these lineages and that these lineages were co-circulating in our cities and provinces. Among the RITM samples, we were able to detect 7 lineages from January 2020 to January 2021. We know this is already local transmission because only 4 of these cases were from patients with travel history outside the country. We also looked into co-infections with the SARS-CoV-2 infection. We found other pathogens in the samples such as Prevotella and Streptococcus. The impact of these potential co-infections still needs to be investigated. Multiple introductions and co-circulation of different SARS-CoV-2 lineages imply that there is a need to strengthen surveillance and control measures at our country's ports of entry. This can prevent the entry and spread of new, potentially more virulent and/or infectious strains into and within the country and to other countries. Only a small proportion of the pandemic has been sequenced. Therefore, we urge caution in the interpretation of data at this stage. Surveillance of known and emerging variants is important in understanding the transmission of SARS-CoV-2 and the implications of emerging variants on disease transmissibility and severity. These 2 research studies bridge the gap between basic research and public health response. Genomic sequencing can provide findings for evidence-based public health policies for the control of SARS-CoV-2 in the Philippines.